Welcome. So let's look at the production of human insulin using recombinant DNA technology. Okay, so first of all, it's important for us to understand how insulin is produced inside the human body so that it will be easy for us to relate it with its production in the, or to understand how it's produced in the body. Then relation on how it's produced using recombinant DNA technology will be much easier. So insulin is produced by the beta cells of the islets of Langerhans in the pancreas. And it is a polypeptide hormone which is responsible for the central regulation of carbohydrates and also fat metabolism in the body. So structure-wise, we're saying it is this protein which contains 51 amino acids. So the active insulin contains 51 amino acids with that molecular weight. Now, it's also a dimer. So dimer, it means it has two chains that are joined together. So these chains are called the A chain and also the B chain. So the A chain and the B chains, these are chains of amino acids that are joined together by a disulfide bond. Okay, so we're saying the first description on the insulin structure was completed in 1954 by Frederick Sanger and others. So the A chain has 21 amino acids, and then the B chain has 30 amino acids. Altogether, this is what makes up these 51 amino acids. Now, these chains are joined by a disulfide bond. So this disulfide bond, this is a bond that is formed between two sulfur entities from the cysteine amino acid residue. So it means chain A has a cysteine amino acids, and chain B also has a cysteine amino acids. And between these two amino acids, this is where the disulfide bond is for joining the chains. Okay, so we're saying initially as insulin is produced in the pancreatic cell, so it is produced as pre-pro insulin, which is 109 amino acids long. Then after that, of these 109 amino acids, 23 amino acids, they act as signal molecules that enable the pre-pro insulin to pass through the cell membrane after formation. And then by the time they're exiting, by the time they're entering the cell, they become 86 amino acids long, which is now called the proinsulin. So from the pre-proinsulin, there's more of these 23 amino acids which act as signal molecules. Then it becomes 86 amino acids long, making the proinsulin. Now the proinsulin is still inactive. That means it cannot act as a regulator of carbohydrates and fats. So this proinsulin is still inactive. It cannot act in the regulation of carbohydrate and fat metabolism. Now, there are some proteolytic enzymes that have to act or to work on this proinsulin by cutting it and exposing its the active side of this proinsulin, and hence converting it into active insulin, which has 51 amino acids. Okay, now that we have a rough, a general overview or an idea on how insulin is produced inside the body, let's look at the production of insulin using recombinant DNA technology. So we're saying the basic step in recombinant DNA technology is similar for production of insulin. So how we produce, so the general production of protein in recombinant DNA technology, that is in part 2 in the previous presentation. It's similar to how insulin is produced, or the steps are similar to how insulin is produced using recombinant DNA technology. Maybe there will only be 1 or 2 parts that will be different. So we're saying initially a suitable vector, which is a plasmid, is isolated from E. coli, and then it is cut open by restriction endonuclease enzyme. So first of all, we need to acquire a suitable vector. So a vector is a carrier, which is the plasmid. So the plasmid is first isolated from E. coli, and then it is cut open using restriction enzymes or restriction endonuclease enzymes. And then the gene of interest, what gene is this now? So the gene that is coding for insulin. So the gene of interest is also isolated from the beta cells and then inserted in this opened plasmid. Okay, so we're saying again, the restriction in the nucleus is the one that is used to identify and cut the insulin coding gene, which is the gene of interest. Then the plasmid and the gene of interest are combined together. So the gene is pasted into the plasmid and then they're joined together by DNA ligase enzyme. Now, this recombined plasmid, which contains the gene of interest from that codes for insulin and that of the then the DNA from the Probacterial E. coli, formed what we call a recombinant plasmid. So this recombinant plasmid is then inserted into the host cell, which is E. coli. So yesterday we looked at how this insertion happens. So this happens by the shock method. So it's inserted back into the host cell, which is E. coli, and now this recombined host cell begins or starts to produce the hormone insulin. So from there on, this is a nutshell. So we know from here that to say, of course, we get the plasmid is introduced back into the host cell, then the host cell. So we know that from the recombinant DNA process, we know that there has to be selection, there's transformation, then there's selection where you put an antibiotic and all that, and then so that at the end of the day, you can only culture the colonies that contain your plasmid. Then after that, you express them. So they're chemically signaled so as to produce the protein insulin. So this is in a nutshell just how insulin is produced. So we're not going into details of that because that was the whole purpose of the previous presentation. Okay, now in 1997, Hakura and colleagues, they chemically synthesized DNA sequence of the insulin for two chains A and B, and separately inserted into the two PBR322 plasmid vector. So this PBR322 plasmid vector. So this is a plasmid, or this is a vector that is used or that acts as a carrier for the genes that code for the A chain and the B chain. Now, so why is this important? This is important because remember we said insulin is made of two, it's a dimer molecule. So it has two chains, the A chain and the B chain, which have to be joined to form the active form, to make the active form of insulin. So the gene, or let's say the gene information, the genetic information that codes for the A chain and that which codes for the B chain, they were identified, the sequence was identified, and then these sequences or then can be inserted, or this gene the gene sequences that code for both the A and the B chain can separately or have to be separately inserted in the plasmid vectors. Which plasmid vectors? The BPR322. So when you look at vectors or carriers in recombinant DNA technology, we'll understand what the BPR322 is. For now, we take it that this is a plasmid that is commonly used as a carrier for the genes that code for the A and the B chains in the production of insulin. So now you have two, because insulin is made of two chains. So we have the gene that codes for the A chain and that which codes for the B chain. Then they are separately introduced in these are the PBR plasmid vectors. So it means one vector is going to produce the A chain, one vector is going to produce the B chain, and then later on they can be joined. So like saying the genes are inserted by the side of the beta galactosidase gene of the plasmid. So this plasmid also contains a beta galactosidase gene. So there's a gene which codes for this enzyme. So next to this gene which codes for this enzyme, that is where the gene which codes for your A or the B chain is inserted. Now, the recombinant plasmids are then separately transformed into E. coli, so there's transformation. The recombinant host produced pro-insulin chains. So first of all, so after that, so you introduce your insulin, the gene that codes for the A and the B chain inside into the plasmids, and you put them next to the beta galactosidase gene. So these are genes that code for this enzyme, which is beta galactosidase. Why this gene? Because the BPR322, they have a high tendency, or they have a high expression. There's a high expression of the beta galactosidase gene. So as they replicate and as they express the beta galactosidase gene, they also express either your A and your B chain. After that, there's production of the pro-insulin chains. What do these pro-insulin chains contain? So the product of this beta-glucosidase gene is fused with the product of the A and the B chain, which is so you have a fused beta-glucosidase A chain and the fused beta-glucosidase B chain separately. The pro-insulin chains A and B are separated from beta-glucosidase by treating them with cyanogen bromide. So let's see using a diagram. It's much easier to understand. Then after that, after detachment, the A and B chains are joined in vitro to reconstitute the native insulin by sulfonating the peptides with sodium disulfonate and also sodium sulfite, making what we call the human. Okay, then we're saying the PBR322 DNA is a commonly used plasmid or a cloning vector in E. coli, which is the strain K12. The molecule is double-stranded, it's circular and double-stranded with that number of base pairs. Now, this plasmid, it contains genes for resistance to ampicillin. By now, you understand why it's important that it should contain an antibiotic resistance gene and also tetracycline. So it has a gene that is resistant to ampicillin and to tetracycline and can be amplified also with chloramphenicol. Okay, so let's look at the plasmid so that it's easier to understand what we've been talking in the 2 or 3 previous slides. Okay, so we're saying so first of all, we have this plasmid. So this is the PPR322 plasmid, which is isolated from E. coli. So it has an antibiotic resistant gene. So here we said it's for ampicillin, tetracycline, but it can also be modified for chloramphenicol. Then it has a promoter gene. So the promoter gene is responsible for expression of these genes, or the gene of interest. So it has the promoter gene, and then it also has the beta-galactosidase gene. Why? Because PPR322 has a high expression rate for beta-galactosidase gene. Then next to the beta-galactosidase gene, you put your insulin gene for the A subunit. So it's just the gene that codes for the A unit. So it means in the next or in the other plasmid, we're also going to do the same thing. So it has an antibiotic resistant gene, a promoter, then the beta-galactosidase gene. Then here you insert the insulin gene, which codes for the B chain. So because we cannot produce both the A and the B chain in one, so it means you produce the A chain in the A chain in one plasmid and the B chains in a different plasmid. So here it says, step number 1, insert genes for each of the 2 insulin polypeptides next to a highly expressed gene, the beta-galactosidase. So these plasmids have a tendency to highly express beta-galactosidase. So you put it next to this gene so that as the beta-galactosidase is expressed by these plasmids, then it also expresses your insulin gene, either the A or the B. And then in step number 2, so as you can note, the plasmids that are carrying our gene of interest are now reintroduced into the host cells, which is the E. coli. So as you can see here, we have the one which codes for the A subunit in red, then the one that codes for the B subunit here, it's in purple. Now, so step number 2, transform E. coli with the recombinant expression vectors and select transgenic cells by the antibiotic resistance. So from the previous presentation, we know that there are those cells, the cells of E. coli that are going to take up our plasmids, and then there are those that will not take up the plasmids. So because the cells of E. coli that will take up our plasmid will be resistant to an antibiotic. So this can be a tetracycline, or let's say this can be ampicillin. So you culture them, so you add ampicillin to the culture. So those that carried our plasmid, they are going to be resistant to this because they carry an antibiotic resistant gene. While the bacteria that are not carrying our gene of interest, or the plasmids that contain our gene of interest, they will not be resistant, and so they'll be eliminated or killed in the presence of these antibiotics. So you transform and then select so that at the end of the day, you only have cells that are carrying our genes of interest. So after that, now so they are grown into a culture or they are grown into high quantities. So you have these bacteria that are going to replicate and produce high amounts of our genes of interest. Then after that, they are expressed. So they're chemically signaled so that they can express the protein. They can express the protein for which they are coding. So that is the function of the promoter gene. So the promoter gene, when it is chemically signals, it results or it initiates the production or the expression of the beta-galactosidase gene, and further also expressing of the insulin gene, whatever gene of interest the plasmid is carrying. So that's why I'm saying, so remember, whenever it's chemically signals, it has high expression for beta-galactosidase to produce this enzyme. So because of that, it will also express your insulin gene. So as the expression is initiated, so beta-galactosidase, the beta-galactosidase protein or enzyme, or let's say protein is produced, which is linked to the A chain. So it will produce the beta-galactosidase product, which will be linked to the insulin gene. So as you can see, the gene here is coded in green. So you have the green chain here representing the beta-galactosidase product. And then you have here in the previous one, the A gene is in red, so which will be the same for, you have a chain which is in red. So the green represents the beta-galactosidase gene product, then the A represents the protein from the A gene. Similarly, so the same thing happens in the in the B subchain. So you have upon expression, so it expresses it highly because it has high expression for the beta-galactosidase enzyme. So there's production of this chain, which is in green, and then there's a purple chain, which represents the B subunit. Okay, so as you can see, then after that now, so after that we're going to write, because now we want the proteins have been produced. Yes, the insulin chains have been produced, but they're still inside the cell. So they are lysed open so that we can collect the A chains. So they are lysed open for collection of the B and the A chains. Now, step number 3, you purify the fusion protein. So fusion proteins, we're saying the proteins from beta-galactosidase and that of A chain, beta-galactosidase and that of B chain, they are purified. So you purify them. Why? Because from this content of the bacteria, there are other proteins that are associated with this. So they are purified so that we only have them in pure form. And then in step number 4, chemically remove insulin polypeptide from the beta-galactosidase protein. So you have the beta-galactosidase protein fused with the A chain, beta-galactosidase protein fused with the B chain. We need to separate them so that we can only work with our A and our B chain. We can work with our A and the B chain. And then earlier we had discussed on how this is happening. So there's chemically removal of insulin polypeptide from the beta-galactosidase proteins. Then after that, it means, so you cleave or you remove these beta-galactosidase proteins, which are in green, and then you remain with your A chain. Now, the A chain, step 5, then combine the polypeptides to produce the functional insulin. So functional insulin is made of the A chain and the B chain. So you have the A chain here after we chemically remove it, separate it from the beta-galactosidase protein, and the B chain, which are then joined by the disulfide bridge. So these disulfide bridge, as we have established that these are formed between two cysteine molecules from both the A and the B chains. Okay, so here we say that another method of insulin production by recombinant DNA technology is designed by Gilbert and a colleague. So in an event where you want to use messenger RNA for pre-pro-insulin, so it's isolated from the islets of Langerhans. So instead of using the gene that codes for insulin, we can just take it from the point where mRNA has already been produced for pre-pro insulin. So this mRNA now have to undergo reverse transcription, and then I'll be inserted into the plasmid, and then the whole process can continue. So whatever we're saying, so in an event we're using the mRNA where you need to reverse transcribe it to form DNA, which is then inserted in the PBR3 plasmid. in the middle of the genes for penicillinase. Then the recombinant plasmid is transformed into a suitable host, for example, the E. coli cell. The host cells produce penicillinase and preproinsulin. Insulin is separated by trypsin treatment. So that is just an alternative to what we had said earlier. Now that we have we have produced this insulin, there has to be a cleanup process. So what is this cleanup process for the insulin which is produced with recombinant DNA technology? So insulin is cleaned up. So here I'll just give an overview. So insulin is cleaned up using the reverse phase high performance liquid chromatography, which uses a non-polar stationary phase and a polar mobile phase. So we use reverse phase HPLC to clean up the insulin that is produced. Okay, so we're saying separation is achieved using an acidic mobile phase. So when we're cleaning up the insulin, we use an acidic mobile phase, normally acetic acid based mobile phase. So at a pH value sufficiently below the isoelectric point of insulin, which is 5.3, in order to keep it in solution. So you need to ensure that the acidic pH is, where it is sufficiently below the isoelectric point of insulin, so that it remains dissolved in solution. The insulin is usually loaded in water-rich acidic mobile phase, followed by gradient addition using acetonitrile. So in general, what we're trying to bring home here is that the insulin cleanup process is done using reverse phase HPLC. So the reverse phase HPLC, it yields about 99% product purity. Okay, and then we're saying the reverse phase HPLC can also be used for polishing. So now in the polishing up of this insulin, so the polishing step, it not only removes E. coli derived impurities, but also effectively separates modified insulin derivatives from the native insulin. So what do we say? So we are saying you can use the reverse phase HPLC to also, let's say, polish up insulin. What is the polishing up? So in the polishing up of insulin, it means it will remove any other impurities. For example, all E. coli derived impurities. Remember, after lysing up, you have other products in combination with your chains. So we're saying, or let's say any other products from E. coli that are still associated with your insulin chains. So we're saying reverse phase HPLC, it can remove all E. coli derived impurities. Apart from that, the other advantage is that it can effectively separate modified insulin derivatives from the native insulin. So we'll see further what modified insulin derivatives is. So they have different characteristic profile compared to the native insulin. So the resultant product after reverse phase HPLC, it has extremely low levels of impurities, which can fail to elicit any immunological responses in diabetic patients. Okay, so what are engineered insulins? Let's look at engineered insulins. What are engineered insulins? So we're saying that engineered recombinant DNA technology facilitates not only production of human insulin in microbial systems, but also facilitates generation of insulins of modified amino acid sequences. So engineered insulins. So the general idea of using microorganisms to produce insulin is that, yes, we can produce the native insulin, so which is the exact type of insulin which is formed by the human body. But we can also engineer insulin. What do we mean? We can also modify the native insulin. How? By modifying or by changing a few amino acid sequences, or by modifying the amino acid sequences of the native insulin, we can come up with the different other forms of insulin, which are known as engineered insulins. So what is the major, or let's say, what is the major aim of generating such engineered insulin analogs. So these are analogs of the native insulin, the normal human insulin that the body makes. However, some amino acid sequences are adjusted, they are modified so that we can come up with insulins that have a different pharmacokinetic profile, for example. So what is the aim of such engineered insulin? So one, it helps in the creation, or it helps in the manufacturing of insulin with altered pharmacokinetics. Engineering insulins, it helps to, manufacture, or present insulins that have an altered pharmacokinetic profile from that of the native insulin, such as faster acting or slower acting insulin. So that's how we have fast acting insulin, slower acting insulin, is because from the native insulin was modified in some amino acid sequences. And then number two, identification of superpotent insulin forms. For example, insulins with higher receptor affinities. It's possible for creation, or it's possible to engineered insulin. They give a possibility, or they give a platform where superpotent insulins can be manufactured. So you modify amino acids here and there such that you come up with a form of insulin that have a high receptor affinity, such that even at the smallest amount of insulin, they would still give an effective therapeutic effect. Then the third one is, we're saying, because of the due to commercial considerations, such as the economic benefits, that would accrue from utilizing smaller quantities of insulin per therapeutic dose. So engineered insulins, they are quite, they're economically effective. Why? Because we'd only have to use small quantities of insulin for a given therapeutic effect. Okay, so let's look at maybe a few examples on, let's say, engineered insulins. Okay, so for example, the insulin amino acid residues that interact with the insulin receptors have been identified. Now, when insulin is administered, which amino acid residues are responsible for interaction with the insulin receptors, and hence resulting in regulation of either carbohydrate or fat metabolism. So those have been identified as A1, A5, the B10s, and up to 25. So what does this mean? So remember, the insulin chain is made of A and the B chain. So where you see A1, A5, A19, A21, that means in the A chain at position 1, A chain amino acid 1, A chain A5, that means A chain amino acid 5. B16, for example, mean in the B chain, the amino acid number 16 has been identified to be responsible for the interacting with the insulin receptors. So these are the specific points in the A and the B chains that have been identified to be responsible for interacting with the insulin receptors, and hence bring about the therapeutic effect of insulin. So based on this knowledge, a number of analogs containing amino acid substitutions at several of these points have been manufactured. So we've altered a number of modifications on these points, because they're responsible for interaction with the therapeutic receptors, have been altered so as to come up with insulins, or so as to engineer other forms of insulins that would have a different pharmacokinetic profile. For example, conversion of histidine to glutamate at B10. So we know that in the B chain, initially there was a histidine. Okay, in the native insulin there's histidine. So conversion of this histidine to glutamate. So histidine is eliminated and replaced or converted to glutamate at the B position. This yielded, or this gave insulin which displayed 5 times activity, or 5-fold higher activity in vitro compared to the native one which had histidine. So that is how it's okay, so we have come up with a form of insulin which is much more active compared to the native insulin. And then several are absorbed from the site of injection into the bloodstream far more quickly than the native soluble fast-acting insulin. So several of these, engineered insulin, they are normally absorbed at the site of action at a much more quick. So the rate at which they are absorbed, there is a much more faster than the native or fast-acting insulin. Such modified insulins could thus be injected at mealtime. So because they are quickly absorbed at the site of action, it's easier, or they provide, they provide an easier wake wake time with the patient, while they can just be administered at meal times rather than administering them, let's say, an hour before meal time, because they can act there and then following administration. Then another example is insulin aspart, which differs from the native human insulin at position B28 where there's proline. So that the proline B28 residue has been replaced by aspartic acid, and hence the name insulin aspart. So at B28, there was replacement of proline in the native insulin by aspartic acid, and hence the name of this insulin is insulin aspart. Okay, so I'm saying this decreases the propensity of individual molecules to self-associate, ensuring that they begin to enter the bloodstream from the site of injection immediately upon administration. So the principle is still the same: alteration of a pharmacokinetic profile. Okay, then the other one was saying the currently used zinc insulin suspensions or protamine zinc insulin suspensions generally display a plasma half-life of 20 to 25 hours. Selected amino acid substitutions have generated insulins which, even in soluble form, exhibit plasma half-life of 35 hours. So the advantage of these engineered insulins is that insulin monologues that have a much more higher plasma life have been generated. Okay, so for those that have a high plasma life, the non-proprietary name of this engineered molecule is insulin glargine. So it differs from the human native insulin in that the C-terminal aspargine residue of the A chain has been replaced by the glycine residue. So in the A chain, the C-terminal, where there was aspargine in the A chain, it was replaced by glycine residue. And in the B chain, of this molecule was elongated at the C-terminus by 2 arginine molecules. So this engineering, this modification, what did they result in? The overall effect is to increase the molecule's isoelectric point, which is the pH at which the molecule displays a net overall charge of 0, and consequently at which it is least soluble. So the pI was moved from 5.4 to a value approaching 7. Another example is Levemir. So Levemir differs from native insulin in that it is devoid of 309 B30. So we're saying at position B30, 309 was removed, and it contains 14 carbon fatty acid residue covalently attached to the side chain of lysine B29. So the whole point of this is that you have the native insulins have been modified at different points to come up with insulins that have a much more better pharmacokinetic profile. So it's either they have better absorption, they have better, let's say, absorption, they have a longer plasma half-life, and all. Okay, what are the effects of engineered insulins? Yes, we can make engineered insulins, but what are the effects of making these engineered insulins? What are the effects of modifying the native insulin into different forms of insulin analogs, that have different pharmacokinetic profiles, that could also have, let's say, different pharmacodynamic profiles. So what we're saying, the alteration of native protein amino acid sequence could render engineered product immunogenic. Maybe we're saying some alterations or modifications of the original or the created or the known, the existing, the nature, the one that exists in nature, the God-made insulin. We're saying it could result or it could render these engineered products immunogenic. And then the other effect is that, so immunogenicity is one effect. The other effect is that these structural modifications of the native insulin, it could also, lead into unintended influences, where you are intending for one effect, but it could also result in unintended and unforeseen influences, such as on the pharmacokinetics or pharmacodynamic characteristics of the drug. So in general, that is it that we're supposed to know about insulin production, that you use two different use two different plasmids, the BPR 322. So we'll see when we look at vectors what the BPR 322 is. So it's this plasmid that is used as a carrier of the A and the B subunit genes that are then formed separately, and then they are joined by they're chemically joined by the methods that have been discussed in here. So this is just a description. This is just a description that shows us the different types of insulins that have been produced in the company. So for example, there's humulin, which is recombinant human insulin produced by E. coli. It's identical to the human native insulin produced by the company, and which so all these are approved, FDA approved insulins that have been produced using recombinant DNA technology. So for example, the engineered insulins, also here you have engineered insulins, Vispro. Okay, so engineered version of native insulin at B28 and B29. This is the company. So it even shows like on the engineered insulins, all these insulins that are here that you've been using, they are all produced by, these are engineered insulins. What does it mean? So you use the native insulin, you need native insulin modified at given position. So for example, in the structure, you have explanations of what modifications were done on the native insulin to come up with these engineered insulins. So that is the best part of biotechnology. It helps us to formulate, or it gives the market, a variety of analogs of insulins, not just the native, but insulins that have a better pharmacokinetic profile, and hence improving patient outcomes. And finally, this is just a diagram that shows the production of let's say insulin. So all in all, that is it. If there are any questions, feel free to write in the comments. If you have any clarifications, feel free to submit and also write in the comments. So don't forget to subscribe so that if new videos come up, you'll be alerted. And also other series are going to continue on biotechnology.